right. Good afternoon, everyone. Welcome to the 8th session of ARCS Forward and the National Scholar Speaker Series. It's so great to see so many faces. Thank you all for being here today. I'm Bethanoni Albrecht, member of the ARCS Minnesota chapter, and I'll be moderating our session today. ARCS stands for Achievement Rewards for College Scientists. The mission of the ARCS Foundation is to advance science in the United States by providing financial awards to academically outstanding U.S. citizens studying to complete degrees in science, technology, engineering, and medical research. The ARCS Foundation has a long history of furthering this mission. Since our founding in 1957 in Los Angeles, ARCS volunteers have been working to elevate young scientists by providing them with unrestricted financial awards. I had the amazing opportunity to be an ARCS scholar during my time in graduate school at the University of Minnesota. I remember being called one evening and offered the award, and I'm pretty sure my jaw hit the floor. They wanted to support me and of course advancing my research, but to have these unrestricted funds was an incredible opportunity. For the next 3 years, I was able to attend conferences, present my work, network far beyond what I would have been able to do otherwise. And it has since opened up this wonderful network here in Minnesota that I'm thankful to be a part of today. And I can say, being on both ends as a scholar and as a member, how valuable this group has been, from exciting engagements in research discussions to career advice and connections. Again, just thankful for what ARCS has provided and continues to provide. I highly recommend, if you aren't already a member, visit our website at arcsfoundation.org, find a chapter near you. During our ARCS Forward session today, we'll be learning about congenital heart disease. We have 2 current scholars doing equally interesting yet different work in the area. Stephanie Helman is from ARCS Pittsburgh, and Omar Troubat is from ARCS Los Angeles. These ARCS Forward sessions have covered a wide range of STEM topics. The ARCS Forward National Scholar Speaker Series was created to present interesting and relevant research topics to both science and non-science audiences and introduce scholars from across the ARCS network of 15 chapters to our members, donors, and friends. On behalf of the ARCS Forward Committee, thank you for joining us today. And as we listen to the speakers, feel free to enter questions into the chat. There'll be time for Q&A at the end. So please welcome Stephanie Helman and Omar Troubat.
Can everyone see my slides? Okay. Well, good afternoon, and thank you, ARCS Foundation, for inviting me today to talk about my research, some of my research findings. My name is Stephanie Helman, and I'm a 5th-year postdoctoral student at the University of Pittsburgh School of Nursing. My research is focused on predictive analytics to learn and figure out a way to quantify neonatal hypothermia burden after open heart surgery. So before we dive into the details of my research, I think it's important for you to understand a little bit of my background and a little information about my clinical research journey. And so I am a trained pediatric critical care nurse. And after I spent some time at the bedside, I went back to school and became a clinical nurse specialist, advanced practice nurse. And I practiced primarily in a high intensity cardiac intensive care unit at one of our nation's top children's hospitals. And there, as a clinical nurse specialist, my primary role was to focus on nursing clinical practice and look at outcomes, measure those outcomes, and determine if we need to make changes. And so it was there that I saw nurses, bedside nurses, reporting unintentional hypothermia in babies after open heart surgery. And so hospitals have robust safety reporting systems, and I would look in those systems, collect data, and implement quality improvement projects. And so if you're not familiar with QI, quality improvement work and frameworks are used by hospitals. And it's a pretty popular methodology to focus on small tests of change on local clinical practice areas. So you can't really extrapolate those changes in other clinical practice areas. That's more leaning into the research arena. But quality improvement work is focused locally. You do small tests of change, you measure pre and post outcomes, and you can see if you can move the needle on any of those things that you're trying to improve. And so I did lots of this kind of work, and I really wasn't making any headway to fix this problem of hypothermia that's not planned in babies after open heart surgery. So I had the opportunity to go back to school and pursue a PhD. And there I knew I needed to learn more about the scientific method because I wanted to take this problem with me, apply scientific methodology, and dig a little deeper. I also knew that we are sitting on gobs of patient data that could assist with investigating and potentially solving this problem. So anytime you're hospitalized and we attach devices or probes to you, we're taking your clinical information and storing it. And some use it for research. And so I'm interested in both the clinical problem, which again is neonates who require open heart surgery and they're prone to postoperative hypothermia, and also innovative data science methods that could help provide us with some insights on how to fix this problem. And so more about unintentional hypothermia. So I'm using this terminology. Well, let's talk about that a little bit. So hypothermia occurs when you lose heat more than you produce heat. You lose heat in 4 ways, and this applies to all ages across the lifespan. So it's not just babies, it's anyone. And so this occurs during 4 mechanisms. You can lose heat through radiation, convection, conduction, and evaporation. Specifically for neonates, they run into trouble because they have more skin surface area than they do have body fat. So they have less insulation to keep in that heat that they're producing. So they lose more heat. That's the risk at baseline for any healthy newborn, actually, not even a hospitalized infant. And so now I was really understanding hypothermia, how we lose heat, these nuanced definitions. And so I went into the literature and I wanted to see if there were other clinicians reporting and other researchers reporting any sort of prevalence of this problem. And so I was targeting first neonates with heart disease. I found nothing. Of course, there are no reports there. It's a very specific patient population. So I thought the next best population to look at would be neonates cared for in neonatal intensive care units. So these are babies that are premature often. They can have a variety of different diagnoses, but often it doesn't include a cardiac diagnosis in large surgical centers because infants or neonates that require heart surgery, they often go to their own cardiac intensive care unit because that care is so specific, especially in the postoperative care period. And so here I was seeing a lot of neonatal hypothermia being reported while in the ICU or during a procedure. And I thought, well, this again is the next best population because although cardiac babies are different in that they have heart disease, cardiac neonates and neonates without cardiac disease, they still share some of the same patient care together. And so this is clinically significant because hypothermia can lead to various deleterious derangements in babies, primarily through impaired cellular function. And so essentially what that means is if your body's thermoregulatory system is off and you're hypothermic, that can lead to impaired cellular function, and that can also then lead to impaired organ dysfunction. And so this is why we're worried about unintentional hypothermia. So more on the population. Congenital heart defects are the leading cause of birth defect associated deaths in neonates. This is the most common birth defect that we know of. There's an estimated prevalence of CHD at birth in the US around 1%, or this equates to about 40,000 new cases per year. Among those, around 6,000 are born with defects that are severe enough that require cardiopulmonary bypass surgery. And this is the population that we're really honing in on. And CHD is very expensive. It costs around $5.5 billion annually, and this constitutes 15% of all costs for pediatric hospitalizations. I want to provide a little more context about this problem and additionally how complex it really is. And so thermoregulation in and of itself is a complex process in the human body. However, healthy babies, you can see in the picture depicted on the left, when you bring those babies home as their caretakers, we are intuitively in tune with the fact that we have to keep them warm. We may not know why, but we know that babies are can easily become cold. And so, as we mentioned before, those mechanisms really are related to them having more skin surface area than insulation, and that provides more of an opportunity to lose the heat that they're producing. And so we do things like clothing them, bundling them, and more importantly, we do skin to skin or kangaroo care, which is one of the most efficient ways to help a baby maintain their heat. But if you look at the picture to the right, this is a post-operative baby. This is what these babies look like after open heart surgery. You can imagine, without knowing what any of that stuff is, you cannot clothe and bundle these babies. You can't do skin to skin immediately in the post-operative period. So this very small, fragile newborn has gobs of tubes, lines, drains, and wires that are really helping them to be hemodynamically stable after heart surgery. And so we have to keep these babies exposed so that we can assess them for hemodynamic instability, which they are at very high risk for in this period. And we also have to allow room for all of these devices. And so that intraoperative period is extremely exposing to these babies, increasing their risk of heat loss. You can't see it in the picture, but we are attempting to keep the baby warm with a radiant heater above head. But that's not the best way to keep a baby warm, but it's the only thing that we have right now. Extremely young babies, we use incubators, but those babies are often not going to cardiac surgery quite yet because they're too tiny. So this is really the best thing that we have so far that allows us to observe patients and also produce some heat. But again, it does increase the risk of heat loss. And so you might ask, so what? This is a common question in research where we try to get to the most important aspect of why you're doing what you're doing. And so we might ask the question, so what? Why do we care about all of this? Well, investigating the complications associated with persistent unintentional hypothermia in the postoperative period hasn't been explored. We know that the first 48 hours after cardiac surgery are the most critical for hypothermia risk. And determining how hypothermia influences complications will inform future temperature management nursing care strategies. So if we figure out how that temperature behaves, why certain babies behave in a certain way, and if those intricacies and individual characteristics influence outcomes, we can change the way that we provide nursing care to our patients. More evidence is needed to fill the gaps. To make this problem even more complicated, there's lack of consensus of what actually defines hypothermia. So some researchers or clinicians use a definition of any temperature less than 36 degrees Celsius, and others use a temperature of less than 36.5 degrees Celsius. So there's a difference of a half a degree. Typically, when we're in the hospital, we measure temperature at a single point in time. And it's much different than how we measure other vital signs. And so we're hypothesizing that single threshold values are not dynamic representations of the complication or the complexity that makes up temperature. And a more robust output is needed to assist clinicians with interpretation of this dynamic indicator of overall health. Additionally, hypothermia has not been linked to outcomes in this patient population. And sadly, most studies examining the consequences of hypothermia in neonates exclude those with heart disease. And so for my dissertation research specifically, I have two aims. One is to identify distinct temporal temperature patterns in this patient population after they've had bypass using various robust analytic approaches. And then we want to take those temperature patterns and determine if they have any influence over important clinical outcomes. And so our design and setting, what we designed was a secondary data analysis of existing observational data, registry data, and longitudinal temperature data collected from the electronic health record. This data registry is called ARCS, and it's a data registry local to the Children's Hospital of Philadelphia. And it includes one of the largest multi-center repositories of neonatal cardiac surgery data available to date, and it's called the Pediatric Cardiac Critical Care Consortium. So our study is being conducted between CHOP and the University of Pittsburgh School of Nursing. At CHOP, they perform around 100 to 180 neonatal bypass surgeries per year. And so our study period is ranging from 2015 to 2019. We're including neonates that are greater than or equal to 34 weeks gestation who have completed their first bypass surgery. We excluded neonates that were gestationally younger than that because they constitute a small percentage of the population, and they also are less likely to go into a bypass surgery because they're too young. So in the end, we procured a data set of 455 newborns. Our time zero for temperature data collection starts at ICU admission after their open heart surgery and continues for 2 days thereafter. Each baby has around 48 temperatures that we've collected. Some have more, some have less. And in the end, we were able to collect 22,000 individual temperature measurements over the 2-day period. And temperatures were measured consistently and continuously with a rectal probe. So this is much better than just intermittent testing. We took all of these temperatures and we pre-processed them into hourly time windows. And this is an example of our data set. So if for any hour there was more than one temperature, we averaged it for that window. If we had missing data, we used the temperatures that were surrounding that hour and applied linear interpolation where it can estimate the temperature and also add directionality to that missing time window. We used group-based trajectory modeling to help us find our trajectories. And so what group-based trajectory modeling does is it estimates the probability of group membership for an individual patient over time, measuring a phenomenon over that time span. And the phenomenon that we're interested in is temperature. And so you can see here that we have 2 images with squiggly lines, and we were able to create or derive rather 2 models. We have a 3-group model and we have a 4-group model where these are distinct. These are groupings of distinct patients. So the patients in each of these trajectories, they're similar in each trajectory, but distinct from the other trajectories. After we determined that there could possibly be a 3 or a 4-group model, we performed diagnostic testing. Both models performed well. However, the 3-group model performed best. So we are going to move forward with the 3-group model. Now, does that mean there's not a 4-group model? I don't believe so. I think there probably is, but we need a little more patient data to test that. So if you look to the left, we'll explain these trajectories. So 36.5 is depicted as a normal temperature for this study. And then we have hours here on the bottom axis. So you can see our group percentage, the group percentages on the bottom of the red trajectory. Around 8.5% of the patients live here. The green trajectory encompasses about half of the sample, and then the blue trajectory encompasses about 40% of the sample. And so that you can see in the red trajectory, these patients were hypothermic for the entire study period, and they never recovered. The patients in the green trajectory were hypothermic in the first 10 hours after surgery and then recovered nicely and maintained normothermia. The patients in the blue group were never hypothermic and they maintained, never hypothermic and maintained normothermia the entire time. So what we're looking at here is around 60% of the patients in this sample experienced some degree of hypothermia during the study period. And this was once not believed to be true. And so this has very important clinical implications once we really start to disseminate and publish these findings. So now that we have our trajectories, we want to know, why are the babies in the red group versus the green group versus the blue group different from one another? And so based on literature, based on clinical expertise, we added certain variables into the model to see if they did pan out as significant risk factors for group membership. Now, certain variables came out that we expected. Gestational age. So if you see, we're labeling this as group 1, and gestational age is here in this row. And if you had a lower gestational age, you were at more risk for being in the hypothermia group compared to the normothermia group. That makes sense. If you had a longer bypass time, this puts you at more risk for being in the hypothermia group versus the normothermia group. Again, totally expected this. If your ICU arrival temperature was low, then that puts you at higher risk of total hypothermia group membership versus normothermia group membership. totally expected that. There's several other risk factors that we have in this model, but you can see in the big red circle, we found something interesting and novel. And so I had a variable in the model that was titled extracardiac abnormalities. So patients that have other organ abnormalities besides their heart defect. And so what we found in group 1, which was the hypothermic group that was hypothermic the entire time, half of these babies had a brain, head, or spine abnormality. And so this increased their risk of being in group 1 versus group 3, which is the hypothermic group. And so this finding warrants more investigation, and I have many plans to pursue that. All of the investigators on my team were all extremely excited about this finding. It opens more questions and more avenues for investigation. So more to come with that. So now that we have our trajectories, we understand why the patients on each trajectory are different from one another. Now we need to look at outcomes. So this is the big so what. So we have to take the trajectories, use them as a predictor, and then determine if they have any influence over outcomes. And how we've defined outcomes in this study, we've separated them out into early onset complications, so complications that occur in the first 48 hours after surgery, and then complications that accumulatively can lead to delayed onset complications up to 30 days in the postoperative period. So some of those early complications that we're interested in would be bleeding, respiratory distress and failure, or hemodynamic instability, all things that hypothermia can influence. And then we also are interested in these longer term outcomes: surgical site infection, impaired weight gain, increased ICU length of stay, and neonatal 30-day mortality. I have not completed this analysis yet, but this is what I'm working on now. Next steps on the whole. So when you're using advanced data analytic methods, it behooves the scientist to use more than one data analytic method. And so we are using k-means or self-organizing maps, which are longitudinal clustering methods, to compare that performance against the group-based trajectory performance. And then we're also going to perform our outcomes analysis, explore the importance of that brain, head, spine abnormality as it relates to hypothermia risk and phenotyping. And then we are actively doing this now. We're working on data collection to apply the models to a second clinical site. So this is underway and data collection is occurring now. And then ultimately what we're trying to do with all of this modeling is continue our search for hypothermia risk forecasting and prediction modeling. So can we predict hypothermia before it's actually going to occur? And then also I am submitting applications for postdoctoral positions. And once I wrap up all of this, we'll start to write my K99 ROO, which is another training grant for postdocs. So with that, I will end by thanking the ARCS Foundation for their support during my postdoctoral study. You can see this picture here is of my 3 children, and the smallest ones were not here during my first few years of study. My son Henry was, but the ARCS Foundation Award helped me to determine that I could manage financially and pursue a study in nursing and research. And so the ARCS Foundation really helped to support some of our childcare expenses during that early time where I had to be on campus full time during very intensive coursework. And so I thank you deeply from the bottom of my heart for your support. And I will end with that. Thank you again.
Thank you so much, Stephanie. Fantastic presentation of your work. Again, we'll wait on Q&A until the end, but as a reminder, feel free to put any questions that you might have in the chat and we'll see what we can get to. Otherwise, I am going to hand over the mic to Omar. It's all you.
Thank you so much. Let me go ahead and share my screen. Okay, great. Thank you so much for that introduction. So I'm Omar. I'm a MD PhD student at the University of Southern California, and I would just like to begin first by thanking the ARCS Foundation for the honor and privilege of participating in this event. And it really is quite, an honor to be here. So for my presentation outline, very similar to the presentation you just heard, I'll start with the background of my journey, how I got to where I'm at. I'll go over some cardiac embryology. So I do research in basic heart development, a lot of the basic science work that has informed a lot of my research interests. And I'll give a background into that. And then I'll talk about my project and then some summary and future directions. So my journey, I'm a Southern California native from Los Angeles, and I'm a first-generation physician and scientist in my family. My father initially immigrated from Jordan in the Middle East, and my mother from Mexico. And both sides of my family, I'm the first person to pursue a career as a physician and scientist. And so that brought a lot of interesting components to my training, and has certainly been an evolutionary process for me to learn what a physician scientist is and how to go about getting there. So I started at USC for undergrad, and I was fortunately accepted into the combined BA-MD program. And what this program is an opportunity for high school students when they're accepted into undergrad to have the privilege really of getting a chance to have a conditional acceptance into the medical school. And as part of this program, they have a tailored curriculum where you can spend time shadowing in the clinic, as well as time in the research laboratory and trying to explore areas that you might be interested in pursuing in terms of a career as a physician or physician scientist. As part of that program, I spent time at Children's Hospital Los Angeles as an undergraduate research assistant. This is sort of when I started my scientific exploration, and it was really bitten by the bug of science, and I really have never left the lab since. But I spent 2 years kind of part-time initially, and then full-time work, and then that transitioned me into the MD PhD program. And I wanted to pursue MD PhD training because I felt, after spending time in the clinic, spending time in the lab, that I was most interested in pursuing a career that allowed for me to be a true physician scientist, to take care of patients, and also to study the ways that we can treat these patients differently and hopefully improve their clinical outcomes over time. And ultimately, my career goal is to become an academic cardiac surgeon scientist. And I have a particular interest in congenital heart disease and patients with these defects. So congenital heart disease, as you heard very well and eloquently discussed, is really caused by malformations during development. And so this is a picture of a normal heart. This is probably familiar to everyone on this call, but to go over briefly how the heart functions, you get blue blood from the body coming into the right side of the heart that then gets pumped out into the lungs. And then that blood comes back from the lungs as red blood with oxygen in it and goes to the left side of the heart and then gets pumped out to the body. And so your heart, one way to think of it is kind of these two pumps, the pump that goes to the lungs and the pump that goes to the body. Well, congenital heart surgeons and pediatric cardiologists have really struggled with how to classify congenital heart disease or malformations in this structure. And so there's been very complex classification schemes. I won't go into it too much other than to say one mechanism you can do this is by anatomic defect. That is, you identify a particular structure, a valve in the heart or a chamber of the heart, and you say, okay, this part is underdeveloped or poorly developed. Therefore, this is the classification for the heart defect. A little more advanced concepts are applied when you try to understand the physiologic classifications of congenital heart disease. So maybe you have an underdeveloped valve, but what is the consequence in the heart? Where is the blood flowing? And so if you have a defect where the blood is moving inappropriately from the left side to the right side, that's one type of classification of heart defect. If your blood is moving inappropriately from the blue side to the red side, those are called cyanotic lesions. And so there was this kind of more advanced classification scheme using the physiology of blood flow in the heart. But more recently, physician scientists have kind of approached the heart with a new lens, and it's trying to look at the heart in the context of its developmental biology or how it formed initially, and trying to see if you can identify the congenital heart defect by looking more closely at the developmental processes that gave rise to that problem. And so this developmental perspective on congenital heart disease is really what informed my experience in the laboratory and my interest as a physician scientist. So again, this is that heart, and this is what it looks like developmentally. So you can see it's a lot more layered and complex than just the kind of the blue-red schemes that we're used to. So when you look at, one of the things that we can point out is that this right ventricle, or RV, which normally pumps blood out to the lungs, is color-coded in yellow here. That is, it's derived from a certain type of progenitor cell or stem cell during development. But you can see that the left ventricle is derived from a completely different population of cells, and that's why it's labeled here in red, a different stem cell population. But what's interesting, and this is kind of the work that our lab is really focused on, is the blood vessel that carries blood from the right ventricle and the blood vessel that carries blood from the left ventricle, labeled here as PA and AO. Both of those blood vessels are derived from the same stem cell population during heart development. And so there is this complexity that's added into the anatomy of the heart when you look at the progenitor or stem cell populations that give rise to it. And together, this PA or pulmonary artery and AO or aorta, that forms what we call the outflow tract. And our lab and a lot of my research has been focused on trying to understand the development of the outflow tract and how to explain abnormalities of the outflow tract or outflow tract congenital heart disease. So one way we can try to understand this, again, applying that developmental perspective, is we look at the outflow tract later, and then we look at it earlier, and we say, how do we get from this earlier primitive structure to the more mature, anatomically correct structure that we're all used to? And so this sort of process of applying the developmental biology to understand congenital heart disease is how we approached my research. And when we look at the outflow tract earlier in development, you can appreciate it looks very different than it does in the mature heart. First, it's one tube as opposed to two. Second, it's much shorter and completely comes from this right ventricle, whereas in later development, you can see it kind of straddles the midline of both ventricles and has two different arteries, as I mentioned. And so when we look at how we can go from the picture on the left to the right, we isolate three steps. First, there is this growth, this elongation, and that elongation allows for this outflow tract to kind of align itself midline. Second is septation or division, where you take the one artery and divide it into two. And third is rotation. When you divide the artery into two, you kind of see that they also crisscross here. And so these three developmental steps, scientists have discovered, have to happen appropriately for you to have a normal outflow tract, for you to have a normal pulmonary artery and aorta. And why do we know that it has to happen this way? Well, when we look at children who are born with outflow tract defects, we can see these problems in alignment, septation, and rotation. So looking first, children who are born that have an alignment problem, where this outflow tract doesn't align appropriately and stays on that right side, they develop what's called double outlet right ventricle, where both the aorta and the pulmonary artery stay committed to that right ventricle and stay on that right side. And so children every year are born with this type of defect, which from a developmental perspective is viewed as an abnormality in alignment. Children who have normal alignment but have abnormal septation or division will develop what's called truncus arteriosus. This is a heart defect of the outflow tract where it maintains that single tube structure as opposed to being divided into two. And third, if you have normal alignment and you have normal septation, but you have inappropriate rotation, you can develop what's called transposition of the great arteries, where your aorta is going to the wrong ventricle, it's talking to the right ventricle, and your pulmonary artery is talking to the left. And so they sort of switched over. And so double outlet right ventricle, truncus arteriosus, and transposition of the great arteries are three of the most common outflow tract defects that we see in children. And we can identify the specific developmental problem that gave rise to that. And so this is that paradigm of thinking about congenital heart disease in the context of the inappropriate development. And so when we look at these three, I kind of said that here, but to repeat it for this purpose is, when you use anatomic classifications, all of these three defects are the same. They're all related to that outflow tract. When you use physiologic classifications, all of these three defects are the same. They all cause cyanosis or sort of that crossover from blue blood getting into the red side. But they're each very different developmentally. And so applying this kind of third layer of analysis allows for us to kind of tease apart these defects and think a little bit more deeply about how we can treat them differently. So background for my specific project is looking at congenital heart disease. And as Stephanie beautifully laid out, that congenital heart disease is, the most common birth defects we see in children, and affects about 1% of the general population. So about 1 in 100 children are born with a heart defect. Also, as Stephanie had mentioned, that these defects can occur on their own, or they can occur in conjunction with some other problem in the body. And she had mentioned you can have brain defects, skeletal abnormalities, and one area of interest for us is craniofacial defects, or defects of the roof of the mouth and the lip. And our group was particularly interested in these children that have craniofacial or cleft lip and palate problems, because we found at Children's Hospital Los Angeles that when we analyzed over 600 children with these cleft abnormalities, we found that they had a 14 times greater incidence of congenital heart disease than you'd expect from just the general population. So these children were particularly at risk for developing heart defects, and that had never before been described. And so when I started in the lab, we had that important clinical piece of information. And the question was, can we take this patient population, these children with cleft lip and palate who are at very high risk for heart defects, and try to understand developmentally what happened, and maybe we can get some insight into how to fix it. So I started and I hit the books, or I guess I hit Wikipedia and the internet, and I read up on the literature, and I said, okay, how can we start to understand from a developmental biology perspective why children with cleft lip and palate can also have heart defects? The roof of the mouth is located up here in your face, and then the heart is all the way down in your chest. How does this happen? Well, it turns out that there's a particular cell type in development, one of these sort of stem cells that I was talking about earlier, and they're called the neural crest cells. And these neural crest cells are very unique cells in that they develop initially by your spine, and some of them will migrate up to your face, giving rise to these structures here labeled in blue. And some of these cells also migrate down into the chest and help to contribute to that developing outflow tract, right? That pulmonary artery and aortic structures we were talking about. So when we looked at the literature, we said, well, these neural crest cells are the only cells in the body that will contribute directly to the face or the roof of the mouth and to the heart. So maybe they have something to do with why children with cleft lip and palate are at such high risk of congenital heart disease. So we looked at those children that at Children's Hospital Los Angeles that had those cleft lip and palate defects, and we compared them to the general population. And so first, looking at this pie chart here on the left, we looked at all the heart defects that you'd expect in the general population. So you can see here the largest components of defects, for example, are things like ventricular septal defects or holes in the heart. And then much more rare, you get defects where you have single ventricle, or one of the half a heart basically didn't form. But when you look at the general population, you have a whole spectrum of heart defects that I kind of alluded to earlier that have different anatomic components, physiologic components, and potentially developmental components. Well, when we looked at the cleft population, what we found is that the heart defects that they had were basically only a few, and they all were exclusively within the outflow tract area. And most closely or most strongly represented or enriched in the cleft patients was what we call tetralogy of Fallot, or double outlet right ventricle. And so this is that defect that I described earlier that's in alignment. And so what this suggested to us is that children with cleft lip and palate, compared to the general population, they don't only have an increased risk of heart defects, but they have an increased risk of particular types of defects. And most significantly, these alignment problems. So maybe there's something about the abnormal alignment in the heart that can help to explain this association. And as Stephanie had mentioned, every scientist will tell you the first thing you need to answer when you're looking into a scientific question is, why is this important, right? What can this information do to really make a difference? And so we looked at these patients and we said, okay, if you have the cleft lip and palate and you have the heart defects, what is that? What is the consequence of that? What's happening to you in the hospital? And what we found is that children who have these both lesions are at significant risk of staying in the hospital longer, staying in the ICU longer with all the complications that were mentioned before with hypothermia and all the problems in the ICU, and also had major morbidity events. They were getting a lot more medication to keep their heart functioning. They were given a lot of medications to keep their blood pressure up. And so, and some of them were at risk of going back into the operating room for more surgery. So this was a critically important question to figure out why children are born with these problems, because there's a real clinical need to try to figure out better ways of treating them. Because the way we're doing it now is great, and they're staying alive, but there's certainly a lot of room to improve. So we took this patient population and we said, great, we have a clinical interest. We want to understand this defect better. We know it might involve neural crest cells, and we think that there might be some sort of common explanation for both the palate and the heart. Around the time we were at this point in the project, there was a paper published by a large genetic consortium, and what they showed us is they studied heart defects and neurodevelopmental abnormalities, problems in the brain development and maturation. And what they showed was kind of like us. They wanted to look at these two different problems in children. They said, if we did gene sequencing, or we collected blood samples from children, sequenced the genes, we found that gene mutations were present in children, shown in this graph here, that were highly expressed in the developing heart and highly expressed in the developing brain. So that is to say, when a child has both a heart defect and a neurodevelopmental abnormality, if they have a gene mutation that can be identified, chances are it's going to be a gene that's going to be both expressed and relevant for heart development and expressed and relevant for brain development. And so it's not likely that you have two separate processes going on, but that there could be some sort of common explanation for these coexisting or concomitant problems. So we thought this would be a great template for us to start our analysis in patients. So we had our cohort and we said, can we look at the gene mutations in these children that have heart defects and cleft lip and palate and see if we can identify gene mutations that are also shared between both of these developmental systems? So that's what we did. So we took parents that were phenotypically normal, so they had no heart defects or no cleft abnormalities, and we took the child that had both the heart defect and cleft palate. And we did gene sequencing. So we collected blood samples from dad, from mom, and from the child. And we looked at all of the mutations in the child that were not present in mom and dad. And so these sort of de novo mutations, we call them, mutations that are present in the kid with the heart and palate problems, but not with the parents, may be some place to start when we try to think about what caused these problems in that patient. And what we found, interestingly, was that a lot of the gene mutations we found are shown here on this table, and they demonstrated to us that these were genes, when we looked into them a little bit more and did a pathway analysis, we found not only were these genes strongly expressed in both the heart and the craniofacial structures. You can see some of that here: cranial skeletal system development, central nervous system development, cardiac chamber development, aorta development, kind of reaffirming what had previously been shown with the heart and the brain. But we looked, and there was one pathway of interest, the non-canonical Wnt signaling pathway, that stood out. And so this genetic data we did suggested to us that not only are the genes expressed in both systems, but they strongly fall onto a single pathway called the Wnt pathway. And so we thought this is a good place for us to start doing a lot of our basic science investigations. And so those are a list of those genes that fell in that pathway. So what is the Wnt pathway? This is a very complicated picture, but I want to show it to emphasize that it's a very complicated pathway and has been studied a lot in embryologic development. But importantly, the Wnt signaling pathway is important for a couple things: for migration and polarity, and for cell growth, survival, and differentiation. These are cellular processes. They basically help the stem cells when they're forming the different structures in the embryo, help them do their job, right? You got to migrate to where you're going. You got to grow and make more of yourselves and help to contribute to some structure. And so when we look at this pathway has been shown to be important in cleft lip and palate and in heart development. And so a previous study specifically focusing on Wnt5a showed that when you remove Wnt5a from the entire mouse, you genetically delete Wnt5a globally, you get a lot of the heart defects that we were starting to see in our patients, these outflow tract abnormalities. And we focused on Wnt5a because Wnt5a is sort of the dominant signaling molecule that drives this entire pathway. And so even though previous studies show that it has some importance in heart development and it has some importance in cleft development, no study has looked at where the Wnt5a signals are actually coming from and how does Wnt5a regulate the outflow tract development. And so you remember the cell that we were interested in are neural crest cells. And so we were wondering, do neural crest cells, right, that one stem cell that goes to the roof of the mouth and goes to the heart, could they potentially be a source of Wnt5a? And this had never before been studied. So we went to the lab, developed mouse models where we started to study this. And so the first thing we did is, this is a picture of a heart on histology. And what we do is we look at the different cells in the heart and we stain for Wnt5a to see where Wnt5a is expressed or what cells are expressing Wnt5a. And what we found here is that these red cells are patterned. These are the neural crest cells migrating into the heart. And you can see that the overlay with where those red cells are also express this Wnt5a. And when we knock out Wnt5a from the neural crest cells, we found that the mice phenocopied or developed cleft lip and palate. You can see here with this white arrow that the whole snout of the mouse is abnormal compared to the normal scenario. And when we look underneath the mouth, we can see that the roof of that mouth doesn't form, very similar to that cleft palate phenotype. And in the heart, what we saw is that these mice developed the same heart defects that we saw in children, that double outlet right ventricle alignment problem. And so just so you can all recall, this is what that looks like when your pulmonary artery and your aorta, both of those arteries are both coming from that right ventricle. That is, they stayed on that right side. They didn't align themselves appropriately. And this is what that looks like histologically, where you have this arrow pointing to, this is the aorta coming from the left side and not from the right. But in the mutant, you can see that the aorta is coming from the right. And so that's signifying this sort of inappropriate alignment. So we wanted to say, what's happening with the cells that are forming the aorta? Why are they not aligning appropriately? And what we found is that the outflow tract, or the part of that heart that forms the aorta and the pulmonary artery, is much shorter in the mutants compared to controls. You can see here in D compared to A. And all those progenitor cells that migrate into that outflow tract to make it longer are all kind of stuck. And so this is how you can see these arrows in panel E show all these cells that are supposed to be migrating into the heart are all kind of stuck back here. They're not migrating the way they're supposed to. In the control scenario, you can see there's a lot fewer of them because more of them are in the heart. And this panel in G here kind of just quantifies that you have a lot less of the migration of this second heart field progenitor into the heart. And this is what the consequence is in terms of that inappropriate alignment, that this, in the normal scenario, you have this really nice curvature of the outflow tract as it starts to align itself toward the middle. But in the mutants, you can see this much more straighter appearance and morphology, showing it's still kind of staying on this right side and is further away from this left ventricle. So it's not moving over the way it's supposed to. And again, just looking at that shortening, here's the normal outflow tract. You can see how long it is. And then here's the control where it's significantly shorter, and that's quantified here. And so the Wnt pathway is important not just for migration, but the cells have to get organized to migrate. And so what this close-up picture shows here is normally you get this really nice single file line with not too many straggling cells. They're all kind of, they know what to do, and they're kind of marching over toward the heart. But in the mutant here, you can see all these clusters of cells kind of bulging against each other and kind of lost. And so the lack of that organization is associated with that poor migration. And when we look a little more closely at the organization, you can see again, here's the normal control situation, very nicely organized compared to the mutant. And you get these really nice projections in the control, whereas in the mutant, it's kind of flattened and inappropriate morphology. And these kind of pictures are showing just some of the markers that lay out how they're supposed to align with respect to one another that are kind of lost in the mutant. And so this was our model. We said, hey, the neural crest cells are providing this Wnt5a signal to the second heart field cells so that they can migrate into the heart, elongate the outflow tract, and form that alignment. And it's this relationship that was sort of the unique one for our research that no one had previously described. And so we wanted to study this a little more closely in the dish in the lab. And so we took those individual cells and grew them on dishes. We created a similar little model where we grew the cells right next to each other so they can talk to each other. And we performed our own migration assays where we drew a little lane in the cells and we watched them migrate into that lane to see how they responded. And then we took some pretty pictures, hopefully. So this was that experimental setup where we grew neural crest cells on the top and these second heart field cells on the bottom. And we knocked out Wnt5a, that molecule that we found. And what you can see here is that in the normal scenario, you have a good migration where it closes off this big lane. When you remove Wnt5a, you don't quite close that off. And when you reintroduce Wnt5a, you can restore that migratory potential. And so we just kind of corroborated and confirmed that mechanism. And these are the nice pictures showing that organization of the cells, where normally they have these really nice projections. They're lost when you remove Wnt5a, and if you reintroduce Wnt5a, you can restore those projections. So the summary of the findings from bedside to bench: we identified cleft patients as a high-risk cohort for congenital heart disease. We uncovered that outflow tracts in particular are in these patients. We identified gene mutations, developed mouse models in the lab where we studied these mutations, and we characterized this mechanism of Wnt signaling as a potential explanation. And back again, how do we go back? Well, the next thing we want to do is we want to validate this mechanism using patient stem cells instead of mice. We want to correlate the genetic data with clinical outcomes, develop diagnostic gene panels for children, and ultimately provide the best care for our patients and their families. And this process isn't just the journey of my research project, but it's also the journey of my own professional journey. Here's me volunteering at Children's Hospital as an undergraduate volunteer in the hospital. Here's me in the lab during my PhD doing the bench research with the mice. And last year here I was performing, helping out with open heart surgery for the first time in the hospital. And so really this bedside to bench and back again is a unique and really a privileged opportunity for me. So lastly, a comment on the ARCS Foundation. The journey of becoming a physician scientist is incredibly challenging. Early exposure and persistent mentorship are necessary, but not sufficient. It really does take a village: family, friends, scientific mentors, clinical mentors. But most importantly, the ARCS Foundation for me has played a critical role in my journey. Not just the financial support, which helps you go to conferences and network and do research, but also the confidence to know that I can be a physician scientist and that this paradigm is an important one to invest in. So thank you to everyone who's helped me along the way. And thank you again for the ARCS Foundation.
Thank you, Omar, for an excellent overview of your work. These presentations are awesome. It makes me miss my grad school days, but very impactful research in a very critical space. And I'm excited to see this continue to advance. So we've got a few more minutes remaining. So there were a couple questions in the chat that I was hoping that we could get to. Stephanie, I was going to start with you with a couple of quick questions here, and maybe they tie together. So will someone be looking at methods for keeping the babies warm? How does this translate to your practice? And then with that, is there a concern as well for the effects of hyperthermia in these neonates?
Yes, so I think that looking at ways to keep babies warm, that's where the good work of clinical nurse specialists and bedside nurses come into play. But first, it's my thought that we have to understand how this temperature is behaving, which is, what my research is all about. And then we need to understand why certain patients are more at risk. So another piece of the research that I think that I really, it would behoove me to focus on would be to evaluate nursing clinical practice that's currently in place. So I do have data that will show in the babies that are hypothermic, I have nursing documentation that can show me how they were manipulating the radiant heaters. And so that's another big problem. Like, we have these devices, but sometimes we don't always use them the way that they're intended to be used. And so I do have to perform another sub-analysis looking at how the nurses were manipulating those devices. So I think we have to answer a lot of questions before we go right to, are there other interventions that we could be doing? But yes, that's probably on the forefront. Second question about hyperthermia. Yes, we're always concerned about hyperthermia, specifically as it's related to bypass surgery. So a lot of patients do become hyperthermic in the 8 to 12 hour window after bypass because they have a huge inflammatory response after they've been on bypass. So we do look for that. We look for hyperthermia because sometimes it can cause arrhythmias in some patients, depending on what surgical procedure they have had. So that's always on the forefront. And then third, we're very concerned about hypothermia after surgery because, cutting into a patient's protective layer, their skin, it puts them at higher risk for infection. So yes, we're always looking for hyperthermia, but not so much hypothermia.
Fantastic. Thank you so much. Omar, I've got a question for you, and this is probably more high in the sky. You talked about that translation from bench back to bedside. And I know there's a lot of work to do. We can't ignore how long that translation can take, but do you foresee that there could be one day an early intervention, like an in utero intervention, that could help increase that signaling pathway and prevent or at least somehow mitigate these conditions?
That's an excellent question. And absolutely, I think there's a lot of interest in developing fetal interventions, not just procedures where you can go in and try to restore flow to a certain area or manipulate, the anatomy and the physiology, but also maybe by encouraging some sort of, medical therapy, some sort of supplementation you can use to help restore, as was mentioned, that signaling pathway to promote heart development at that critical time. But one of the interesting things is that in addition to the initial loss of that pathway causing the heart defect, what we're finding now, because of great surgery and great clinical care, these children are living into adulthood now. And so the question that's come up from our work is, does that loss of that pathway maybe create opportunities for you to intervene now when they're children? And maybe they're more prone to developing heart failure or more prone to doing poorly after after heart surgery. How can we use that developmental perspective to now intervene, hopefully in utero, but if not, also later on in life, because now these children are living there and are going to need help and care for the rest of their lives? Great question, though. Thank you.
And actually, so a follow-up with that, with the potential of the in utero or the fetal intervention, how early, I'm not sure if you have data on this, but how early in the development would this need to happen?
It's a great question. It would need to happen in the first trimester. So the heart development happens very early in the body. And there's some study about whether or not you can have a later intervention and maybe make a heart defect less severe. And that's currently ongoing. There's some groups from Boston Children's Hospital that are trying to figure out how to intervene later in the second and third trimester and lower the severity. But if you want to absolutely cure heart defects, you'd have to intervene very early in the first trimester. So this would certainly be something in the context of, again, some sort of in a prenatal supplement type of phase, or maybe in a planned pregnancy where you can kind of say, my first child had a heart defect, how can we go into planning this for the next one? But maybe intervening a little later and lowering the severity would be clinically beneficial as well.
So Omar, I know my daughters both have had 4 pregnancy between them, and they did, ultrasounds really early looking at cleft deformities. And is that why they're doing it so early? Because
Absolutely.
It was definitely early in the pregnancy.
Absolutely. You want to try to identify it early so you can plan what you can do as soon as the child is born to try to, first during, in utero, if there's anything you can do to help promote the growth of the child. And then certainly as soon as they're born in that neonatal period, that early post-natal period, if you can try to intervene and help them grow, help them, making sure that they can kind of have, as great an outcome as possible. So you're absolutely right. They do look early, and they look early because of research like this that says you need to look early if you want to have an important clinical impact.
Well, I also had 2 children with hypoplastic left heart, and that was in the 80s. And so they were looking at that too, but they said they couldn't determine that early in the pregnancy.
Yeah, that was actually one of the examples I was giving of the Boston group trying to intervene a little later in that. That's one that you get a sense maybe early that it might develop, but you really don't know until a little later in pregnancy. So that's a different heart defect that that is currently being studied as well.
And that's where our second son had surgery, was Boston Children's. We went
Wow.
from Atlanta to there.
That's incredible. Thank you
Yeah.
for sharing that.
And I know we're right at time, and I want to be cognizant of everyone's schedules today. But thank you both, the speakers. Fantastic work, fantastic presentations, incredibly important. Exciting to see where it's going to go. So thank you all for participating in this session today. We'll send out a link so you can share it with all of your friends and family, because again, incredible work. And there will also be a survey. So remember to fill out the survey. We do use that information to help plan our sessions. Very useful for us. And I want to make a plug really quickly for the next session. So the next one is going to be May 18th at noon Eastern. The topic will be oceanography, and will feature Jess Perlman from ARCS Hawaii and Mia Sanko Casey from ARCS Oregon. Thank you all again for attending, and we really look forward to seeing you in May.